Hi everyone, and welcome to this Copernicus Marine Service training for ocean color. My name is Silvia Pardo. I am an Earth observation scientist at the Plymouth Marine Laboratory in Plymouth, United Kingdom, and I'm here today to help you explore some of the ocean color products you can access through the Copernicus Marine Service. This is part 3 of a 3-part series in ocean color, so please feel free to go back to part 1 if you need a refresher on data structure and opening, extracting, and plotting data. During this third and final session, we will use Copernicus data to explore the optical variables distributed in the ocean color from satellite observation products. So in this exercise, we will study a region where a variety of large European rivers reach their end, and we will try to find the optical signature of the river inputs in the changing color of the seawater. A quick reminder that we are going to be working in this Jupyter notebook, so as I'm sure you know by now, you can execute the cells by pressing the play button and can stop the execution using the stop button. Before getting our hands into the optical data, maybe let's try to understand very quickly a little bit more of the physical principles behind ocean color. And as we discussed in part 1, water molecules absorb almost sunlight except for the blue part of the spectrum, which is reflected back. And this is the reason why open ocean region, where the ocean is relatively empty, looks deep blue from space. So then we have different types of molecules in the water sometimes, and the presence of those known water molecules alters the behavior of sunlight as it penetrates the ocean surface. So in this figure we have some examples of one of those water components. And the effect of the suspended and dissolved organic or inorganic matter basically depends on how big the molecules and how many of them they are. And when the light interacts with them, two things can happen. So radiation can be absorbed as energy, or radiation can be scattered back and then be detected by a sensor in a plane or in a satellite, or in general a remote sensing sensor. So as we discussed in the previous exercise, phytoplankton contain chlorophyll, a pigment that absorbs mostly the blue portions of the sunlight to produce carbon for photosynthesis, and it absorbs a little bit less in the green side of the spectrum. So we have focused on this chlorophyll for exercises 1 and 2, but in this particular exercise, in this final exercise, we will look for a different type of molecule, in particular suspended inorganic matter, which is frequent in coastal areas and shallow waters and contributes to the ocean color in the green and the yellow and also in the red parts of the light spectrum. So let's see an example of these water constituents in this natural color image acquired on the 16th of April 2014 by Moody's on board of NASA's Aqua satellite, and this captures the most of a variety of large European rivers as they empty into the northwest shelf seas, and you can see that both the river and the river banks appear in light brown, which we can easily identify as like a mud type sediment, and we can study the shape of the sediment plumes and their persistence to measure currents in the area, for example. So in particular, we will be studying this, which is called the Anglian Plume, which is one of the largest features in the northwest European shelf. So this extends from the Thames estuary across the North Sea towards the Danish coast, and the South and North Sea generally experiences the highest suspended particulate matter concentration, followed by the English Channel in here and the Irish Sea, which is outside this picture. And in contrast, the off-shelf Atlantic waters of the southwest are the clearest, so they don't have that much suspended matter. And these suspended particular matter concentration, especially along the coast, as you can see here and in here, is usually at its highest during winter, because we have more precipitation, and also we have there's a lot of resultant river discharge due to this high precipitation. We also have events that have to do with the meteorological events, like storms and strong winds, and all these processes are strongly modulated by the influence of tides and waves. So this is the motivation for the exercise. Let's start coding. As we did in part 1, we will set up the environment and data paths. We will use the following libraries. All of them have already been used in parts 1 of 2 of this ocean color practical session, so I won't delay you in there. Once the environment is set up, we will define the optical data set. So as we saw in the presentation, the ocean color tag produces a variety of optical products, which I have listed in here. So in this first section, we will look at the spectrally resolved remote sensing reflectances, and these variables provide critical information regarding the visible radiation reflecting from the ocean surface and detected by the sensors after passing through the sea-air interface. So while solar radiation can be seen as a continuous variable, basically as a continuous spectrum of light, at sensor level it gets some sample as a subset of wavelengths, a subset of remote sensing reflectances. And this means that remote sensing reflectance products are only available at this specific wavelengths, namely at these wavelengths, or wavelengths equivalent to those expressed in here. So every band is distributed on an individual file labeled with the corresponding band center wavelength. As you can see in here, this file, for example, is for the band that is centered at 443 nanometers. And these wavelengths match this CWIP's set of reference for remote sensing of reflectance, and CWIPS is the ocean color sensor of reference for the reprocessed data sets. So these data sets in here are slightly different from those that we have used in parts 1 and 2, so I will refer you to the documentation if you want to know more. But as you can see, the naming is quite similar, so basically the only difference is in here, and the fact that the resolution is slightly lower than the files that we use for the chlorophyll dataset. So as we did in part 1, we need to define the path to the data files. So to do this, we define the usual root directory, data directory, and results directory, which as always you need to modify accordingly if you want to use data that is hosted in your own computer. Then we define the file names that are going to be relevant for us for this first part of the exercise, and we open them using the open dataset function within the Xarray module, and then we load some of the variables. So for example, in here we are already loading the latitude and longitude from the remote sensing reflectance at 443 nanometers dataset. So now we have a bunch of remote sensing reflectances. So for example, in here we are loading another 3 variables. What we can do with them basically. So the first and easiest thing to do would be to map them. But plotting a new variable might be tricky. We don't know anything about the types of the data, the ranges. Is this a linear variable or is it log normal as a chlorophyll? So to answer this question, we can explore the data structures as we did in part 1 and 2. So you can try this on your own in here. Just pause the video and try to access the different attributes, the dimensions, access the actual variable descriptions inside, so you can see the units, the range of values, and you can have more information about all these questions that I have posted in here. And you can also try and look at the spectral characterizations of remotions and reflectance in the literature if you're interested to, again, know how these things are measured, what kind of values they have. Regarding the literature, for example, the values of remotions and reflectances data across the spectrum are dependent on the water constituents, and there have been many studies trying to characterize the typical remotions and reflectances spectra for different water types as characterized in here, for example, by Jackson et al. 2017. You can find the reference by clicking on the paper. So in general, spectra in the shapes of water classes 1 to 7 will be obtained for clear open ocean waters, and those showing a spectral line similar to classes 11 to 14 will belong to more turbid coastal waters. So if you have information about which optical water type you're looking at, you will have information about the range of values each one of the remotions and reflectances at each one of the wavelengths have, basically. So for example, remotions and reflectances in the blue are around 443 nanometers, so around here. Only reach values just above 0.01 for the very blue oligotrophic waters, as one will expect in the North Atlantic open seas. So, going back to our variables, to understand how the different wavelengths and by extension the different water constituents contribute to the ocean color, we will first try to build a natural or RGB, red, green, blue color map of the area. So for this example, the red will be band at 670 nanometers, green will be the band at 555 nanometers, and the blue one will be the band at 443 nanometers. And as I said before, we loaded in there. And then we, in the first approach, we have a look at the three bands separately. So by now you're obviously an expert in plotting, so I won't go through the whole plotting sequence. It's exactly the same as we used in parts 1 and 2 for the chlorophyll. The only difference is that I've used a slightly different color map for the three exercises to characterize the blue, the green, and the red remotions and reflectances. So for this specific color map, which is the remotions and reflectance at 443, and we are looking at the 2nd of April 2013, the lighter the color, the larger the amount of blue light, blue in the 443 nanometer wavelength, our sensor is detecting. So in particular, the Anglian Plume that we were talking about earlier is particularly light in the map, so we can say that the signature of the river plume is visible at this wavelength. And we can compare the blue band with the green and the red. Again, a similar or practically identical plotting sequences using the green and the red color maps for the green and the red bands. You can now visually compare the three of them, the blue, the green, and the red. And if we do that, it's clear that while the sediment plume is visible in the three bands, it's visible in the green, and is clearly visible in the red as well, the effect of the river plume is more intense in the green and red bands than in the blue band. And this is due to the optical characteristics of the particles released by this river and the effect of this input in the ocean color. And now we can try to combine our three channels in on a RGB image, so red, green, and blue image, using the image show function. So this is a different use of the image show that we will, the one that we saw in the part 1. And there we had only one data variable, so we had to convert our chlorophyll data from 3D array to 2D array. But in here we have three data streams. Remember that we have one band for the red, one band for the blue, and one band for the green. And this naturally describes an RGB model where an Rgb model is an additive color model in which red, green, and blue bands are added together in various ways to reproduce a broad array of colors. So one color in the Rgb color model is described by indicating how much of each one of the red, green, and blue is present, and the color is expressed Rgb triplet, so 3 numbers, and the values for each one of the components can vary from 0 to a defined maximum value, which is usually 1. So all this means that we need to normalize the reflectant values into Rgb values, which must be between 0 and 1, and we can perform this conversion by normalizing the 3 channels, so dividing each array by the sum of the 3 arrays, which is what we'll be doing in the sequence in here. So basically, we are just interested in this box of data. As we did in sections 1 and 2, we have a way of zooming in into the data, so extracting only the pixels inside this box. So we've done this before. The part that is new basically is that in here we are defining the total signal, the total Rgb signal, by adding the red, the green, and the blue, the same sum in variables. And then in here we are normalizing. So as I said, we are dividing the red signal, remote sensing reflectance signal, by the total signal, and we are clipping the values, or normalizing the values to be between 0 and 1. Then we have some code in here that you might or might not want to implement. So first, this piece of code that shows you how to implement a gamma correction, which is usually applied to control the brightness of an image and make the image not look too dark. Might not be needed, but I thought it was a very useful piece of code and very simple, so I left it in here commented, so you please feel free to investigate. In the same line, we can calculate a true green band from the other 3. This again is by no means essential for the exercise, but takes into account that sometimes we need to work with sensors where the green channel does not measure the actual visible green light, but the near infrared band sensitive to chlorophyll instead. So we could use that channel in place of green, but it will make the green in our image appear too bright. So instead we tone it down by interpolating the value to simulate a more natural green color. So this is what we're doing in here. And finally, we have to stack the red, the green, and the blue channels to create a single straight dimensional Rgb array. So as usually, this function gets the array list, that basically the list of arrays that we're gonna aggregate, and the index for the new axis as arguments. Again, we've used the stack function before. Final operations using numpy, nunt, and num. So this function over here, converting nonce to zeros, so they show as part of the cloud mask. And last but not least, using the usual plotting sequence, but without the p-color mess, we use an image show function instead. We visualize the data. So we are visualizing this stack Rgb data. So this is the resulting map, which is very coarse because, as I mentioned, the resolution is lower than the chlorophyll maps, but it does provide a lot of relevant information about the types of water we are seeing. So open waters, for example, in here are pictured in blue or bright blue. Then we have coastal areas which have like a light brown, light yellow tint, and then we have areas with high sediment discharge, such as the river mouths, appearing in dark brown. So to finish this subsection, we will try to extract the reflectance spectrum for the Anglian Plume, to see how the resultant spectral curves match the different water types we saw in the Jackson et al. 2017 water type graph that I showed above. So to do this, we will use the green band at 555 to to define the extent of the river plume. So this is exactly what we are doing here. We're using the green band at 555 nanometers, and we impose a threshold. So everything, every pixel with remote sensing reflectance at the green band higher than 0.025 stereo radians to the -1 belongs to the river plume. So before that, obviously, we will subset the data by identifying the array indexes for the box we are interested in. As I said, I define the river plume, and once the plume is localized, then we define the list of wavelengths that we are going to extract over there. We initialize a remote sensing reflectance empty array of the same shape as the wavelength. So basically we will have one-dimensional array with one element per wavelength. Then we loop over the different wavelengths, extracting the plume values and calculating the median value as we go. So for each one of the wavelengths, we identify which position the wavelength has on the wavelength list. We use that information to select the right file name and variable names, as you can see here. Then we open the file, then we load the variable, then we extract the part of the variable that is inside the plume using, inside the box using the geographical subset indexes. Then we select within that box the pixels that belong to the river plume using the river plume indexes. And finally, we calculate the median of the remote sensing reflectance, and we store it in this variable. In this variable over here. So this is the calculation, the variable that hosts the median, and then we assign that value to the remote sensing reflectance element in the index position. So if we print the final list, you can see that the resulting spectra for all those pixels that belong to a river plume have a really strong signal coming from bands at 490 and 455. So those two values are the highest of the time series. So we can easily do the same for the remote sensing reflectance value ranges that might be defined in all the water types. So for example, in this second box, still using 555, the green band as a reference, we can select the locations where the green band values are between this range, so between 0.001 and 0.0025. These values are usually correspond to coastal or shallow waters. So we select those indexes, we initialize an empty array with the same shape as the wavelengths, and it's exactly the same sequence, but just for the pixels that belong to the coast or shallow waters. So we loop over the wavelengths, extracting and calculating the median value of the coastal values, ignoring masked values as we did before, and we store the value in the coastal remote sensing reflectance array as we are doing here. And finally, we print the remote sensing reflectance coastal array, and we can see the strong little signals are now coming for bands 443 and 490 over there. And finally, we can still, using the same band, extract pixels for the clear waters that are further north of the river plumes, and we will define those as remote sensing reflectance values below 0.001. So again, we loop over the wavelengths, extract, we load, we calculate the median value for these indexes in here. So we extract those and we calculate the median for the open or clear water values. And finally, we store it in the clear water remote sensing reflectance index. And we print the resulting spectrum, and we can see that the strongest signals are still coming for bands at 443 and 490 nanometers, but the average values for these bands are lower than those for the shallow waters spectra that we just see above. So at this point we have mean remotency reflectance values per band for 3 regions: the river plume, the coastal and shallow waters, and the open ocean or clear waters. And we can interpret them as spectral lines characterizing the very different processes that are going on in the region. So to see that more clearly, we can plot them as follows. So we have used all these functions already. So we have 3 plots in different colors, in yellow and red and blue, and each one of them is the spectral line for these particular regions. So as an exercise at this point, you can now compare this plot over here, so the spectra for these 3 regions for the 2nd of April 2013, with the water classes that I showed you that came from a paper, from the literature, and try to decide which class does the clear water spectra much better, and the same with the coastal and river plumes. So that's the first half of the session, and for this second and I think final part of the session, we will have a deeper look at the optical signature of the European rivers. So we just saw one. Here's a map of the many of them that have a strong optical signature, or have a strong effect in the color of the ocean. And we will do this by looking at the most sophisticated optical products in the catalog. So that's the suspended matter, SPM, the Secchi transparency depth, which is called SD in the catalog, and the diffuse attenuation, so that's KD490. So while the spectral sampling over here that we have just performed can give us a good idea of the character of the water, this analysis needs to be combined with other variables and a full model of the in-water light field to be able to identify inorganic matter and retrieve its actual concentration. So a measure of the concentration can be derived from chlorophyll concentration and radiance values at 555 and 570 nanometers. And in the same way, the absorption and back scattering coefficients can be obtained for reflectances, and these can use to calculate the value of the diffuse attenuation coefficient. So all those variables that we've seen until now, the chlorophyll and the remotency and reflectances, are being used together to provide these optical, very sophisticated, high-level optical products that we are going to see in this section. So let's have a closer look at these products from satellite passes acquired over the area for the same date that we have been using so far. So that's the 2nd of April 2013. This is a period, as we saw, with where we had a high sediment depositions events. So we will start with listing the relevant Siemens files and loading the corresponding variables. So that's exactly what we've done in here. File list, opening, accessing the daily latitude and longitude. We only need to do this because the gradient is exactly the same for all the 3 files. And finally, we load the variable we're interested in. We will first have a look at the suspended particulate matter product, so SPM. Basically, we will use exactly the same plotting sequence. By now, there's nothing new apart from a different color map, and this is the resulting SPM suspended particulate matter product, which is measured in grams per cubic meter. So the highest concentrations are observed in coastal areas, so particularly in river mouths. As you can see, there's the plume that we saw in the first half is quite visible, and we can see the signature with SPM values above 20 grams per cubic meter. So because SPM contributes to water clarity, through light absorption and scattering. The concentration determines the amount of light available for photosynthesis in the column. So there is a high correspondence between this kind of map and the chlorophyll map. And if you're interested, I will encourage you to look into that. So SPM concentration cannot only be used to detect the extent of the plume, as you can see in here, but also as a measure of the river stream flow and how much sediment it yields. So this will allow us to draw comparisons between different riverine systems, and this particular riverine system and the associated East Anglian Plume has been well studied by various research groups. So I believe I've left you a paper, a reference to a paper somewhere, probably further down, to a recent paper 2019 by Greenwood et al. That estimated that this river plume, the point of maximum gravity in the SPM had a value of 25 grams per cubic meter, so above this yellow value in here. And then they used this value as the SPM threshold to measure the extent of the area influenced by the plume, resulting in an area of 5,724 square meters. So as the next exercise, we will do exactly the same. We will use the index selection tools that we've been using for this whole exercise, together with the SPM product, to define the boundaries of the plume. So we start by defining a box around the plume and use the index selection tools to extract the zoom in SPM array and latitude and longitude array. So this is the interesting bit where we use a 20 grams per cubic meter threshold over the SPM value to define the pixels that belong to the river plume, so those with SPM values above 20 grams per cubic meter. And then we obtain the indexes of the plume and we store it in this variable. We create an empty plume array in the same shape as the SPM zoom variable. Then we flag those pixels, changing the values from 0 to 1, exactly as we did with the phytoplankton plume in part 2. And finally, we plot the extent of the plume as defined by the plume variable. So the plume variable is here. We have just assigned the values of 1 to the plume indexes, and then we can plot it in here, which again, as the RGB map is a little bit too coarse. Also, as the plume flag array has only two values, 0 and 1, using a full color map doesn't make any sense. So instead, as we did for the phytoplankton bloom, we have built our own color map with only two colors, white and red. So the pixels in white are those that are basically clear waters, so to speak, and the red pixels are those that belong to the river plume. And from here, basically, after plotting, we will try to get a measure of the geographical extent of the plume. So we take advantage of the regular space latitude and longitude grid to derive the spacing between latitude and longitude pixels or grid points. So that's it. Then we perform a really rough approximation to translate for coordinates to distances. So the details here are beyond the scope of this session, but just to explain what each line is doing. Here we are converting latitude values from degrees to radians. Here we are building an array for pixel widths in kilometers. And finally, here we define an array for pixel areas in square kilometers. So from here, it's basically we're quite straightforward to calculate the extent of the plume. So we extract the pixel area values in here just for the pixels that belong for the plume. Then we only need to add them all, like in here, using the numpy sum function to generate the plume area in square kilometers, and then we print it. And finally, we can have obtain a rough measure on the intensity of the plume by using the numpy amax function, as we are doing in here, as you can see in here. And then we print the results, and all this is resulting in a plume area about 6,670 square kilometers, and a maximum SPM values or 100 grams per cubic meter. So that's a lot. At this stage, you can again take a short break and maybe go read the paper I've referencing here and see how these values compare with the ones that were obtained in the paper. Now that the plume has been clearly identified, we can have a look at the other optical variables. So for example, in here we will look at the Secchi depth, which can be defined as the vertical depth at which opaque white disk, which is called the Secchi disk, stops being visible from the surface. So let's see what this product looks like for this date. So that's the, again, 2nd of April over the English Channel. Same plotting sequence as for SPM, but using a different color map. And this is what it looks like. As with SPM, river discharge, as you can see here, can cause huge perturbations in water transparency. So we have Secchi depth values as low as 2.52 meters within the plume in this particular period. And this means that the Secchi disk, if deployed within the plume, we will only be able to travel vertically for 2 meters before completely disappear from sight. So in some cases, the Secchi depth can be interpreted as a proxy for the trophic state of the water, so how productive the water is. And we can also derive water quality indicators from the Secchi depth. So Secchi depths lower than 5 meters can indicate highly productive waters, and the water quality falls to the very low category for values lower than 3 meters, which is obviously the case for this river plume. And last but not least, we will have a look at the diffuse attenuation. So that's KD 490, which is a proxy for the turbidity of the water column. So the vertical visibility, so that's the Secchi depth, is inversely proportional to KD 490. So the higher the turbidity, the lower the vertical visibility, which is logical. So let's see what this product looks like for the date in the English Channel. So river discharge again and sediment fluxes around the area have a stronger effect in water turbidity. So we have values of turbidity reaching 0.2 meters to the -1 in some of the most turbid areas. So this means that the concentration of scattering particles in the water column is very high, and light gets quickly diffused after penetrating the sea surface. So in other words, there's suspended matter that's blocking the path of the solar radiation through the water column. As with SPM, as I said when we were looking at SPM, light availability is an important driver in the growth of many subaquatic organisms. So high turbidity events can be quite detrimental to the ecosystem. So in many instances, there is a significant, as I mentioned, relationship between the chlorophyll and the optical products. So as a final exercise, I would suggest that you download the corresponding chlorophyll file for this date and build a map of the chlorophyll concentration to study the character of this relationship. And with this, we have reached the end of this Copernicus Marine training for ocean color in the Northwest Shelf Seas. I really hope you have enjoyed the practical sessions as much as I have enjoyed preparing them. Hope these exercises have triggered a lot of questions and ideas that we hopefully will be able to discuss during the debriefing session. It's been a great pleasure to help you explore some of the ocean products you can access through the Copernicus Marine Service. Take care. Thank you. Bye-bye.